Hello everyone, welcome back to my YouTube channel Floral Biocanvas. As in previous video we have studied about microsporogenesis where we saw the formation of microspore. Now we will be seeing in this video about megasporogenesis, that is formation of haploid megaspore from diploid megaspore mother cell. So here there is a megaspore mother cell which undergoes meiosis division. After meiosis it will be forming 4 megaspore cells which will be haploid, and these megaspore cells are arranged in a linear fashion. So we see that there is a linear tetrad of 4 haploid cells, that is megaspore. Now among these four cells, 3 of them, upper 3 cells will degenerate. This upper 3 cells got degenerated and the lower one remains functional. So there is a functional megaspore. The functional megaspore undergoes 3 successive free nuclear mitotic division. It means that the one single functional megaspore will undergo 3 mitotic division. After 3 mitotic division, it will be forming 8 nuclei, and from this 8 nuclei, 4 of them are arranged at each pole. Means 4 nuclei will be present at one pole and the other 4 nuclei will be present at the another pole. So from these 4 nuclei which are present at one pole and the another pole, one will migrate towards the center. Means from one pole, the one nuclei will migrate towards the center, and from here one will migrate towards the center. So at the center there will be 2 nuclei which are termed as polar nuclei. Now let's see there the nuclei present at the micropylar end. Micropylar end, which is a opening which gives entry to the pollen tube. The nuclei, 3 nuclei present over there are termed as egg apparatus. It is having a haploid cell at the center termed as haploid egg, and there are 2 haploid synergid cells. Okay, so synergids have a filiform apparatus which is hair-like projection, and it gives guidance to the pollen tube towards the egg so that there will be fusion between the male gamete and the egg. Like if we see that there is a synergids, it is having filiform apparatus at the center, there is a egg, and this filiform apparatus helps in guidance of pollen tube towards the egg. This is filiform apparatus. After this there is presence of antipodal cells, 3 antipodal cells which are present at the chalagial end. The upper end, that is chalagial end, there is present of 3 cells that are termed as antipodal cells. Now the 2 haploid polar nuclei which were there, these nuclei fuses to form secondary nucleus. So we can see now there are 3 antipodal cells which are arranged at the chalagial end. The polar nuclei fuses to form secondary nucleus. There is a synergid cell with the filiform apparatus, and there is a haploid egg cell. So this is the embryosac which is having 7 celled and which is 8 nucleated. How 7 celled? So 1, 2, 3, 4, 5, 6, 7. There are 7 celled means 7 compartments, and 8 nucleated. It means that there are 1 nucleus, 2, 3, 4, 5, 6, 7, 8. So it is 8 nucleated. The kind of embryo sac development here, it is monosporic development. It means that the embryo sac developed from single megaspore cell. When we saw that there were, after meiosis we got 4 megaspore, but among them 3 got degenerated and 1 remained functional. So it developed from single megaspore. Therefore this kind of development is known as monosporic development. So let's see the female gametophyte. It is colorless, endosporic, and it is concealed in the ovule enclosed by ovary. The diagrammatic representation of this is like we saw there was a single megaspore mother cell which was diploid. It undergoes meiosis division to form 4 megaspore haploid cells arranged in a linear fashion, among which the 3 got degenerated and 1 remained functional. This functional megaspore cell will undergo 3 successive mitosis division. So this functional megaspore, after mitosis 1, it will be forming 2 nuclei, and after mitosis 2 there will be 4 nuclei, and after mitosis 3 we can see there will be altogether 8 nuclei. 4 of them are present at one pole and the 4 of them are present at another pole, among which like the one nuclei will migrate towards the center and one will migrate towards the center. They will be forming polar nuclei. Okay, so we can see that there is 3 antipodal cells which forms the antipodal cells which is present at the chalagel end, and these 2 polar nuclei present at the center fuses to form the secondary nuclei. There is a haploid egg cell with the 2 synergid cells with filiform apparatus. So this is kind of mature embryosac, that is female gametophyte, which is 8 nucleated and 7 celled. Okay, so this was all about today's video. If it was helpful to you, please hit the like button and subscribe to my channel for more new videos. Thank you.